Okay, good morning to everybody, and thank you for being here. Welcome to the Metabolomics workshop. Today in day one, we will focus on understanding the foundation of metabolomics research and profiling by mass spectrometry. And particularly during the first 2 hours, we will focus on understanding, one, the foundation, two, the main bioinformatic workflows for performing untargeted metabolomics. And during the last hour, we will work together with real experimental data to extract metabolomics information from untargeted studies. At day 3, as I briefly mentioned before, there will be a class test. This will be regarding the topics of day 1 and day 2. The test will be 1 hour, 14 questions, open answer and multiple choice, but also one hands-on question, which will be time restricted. And we will not have access to AI or AI on phones. So any questions so far? Okay, good. So why metabolism? Why systems biology of metabolism? I like starting my presentations with this stylization of the Vitruvian Man by Leonardo da Vinci. And the reason why I do so is because I do believe that throughout the history of science and research, this was probably one of the first attempts at science looking at our system from a holistic perspective. In this case, Leonardo was trying to understand the geometric proportions of the human body, right? Whether there is some underlying principle that was connecting the proportions of the human body. But it's interesting because he was one of the first one really at trying to understand us as a system, as opposed to looking at the disconnected parts, which is really what we do today in our research. How does this system work? And particularly, what is the role of metabolism in health and disease? And also, what is the activity of these metabolites? And can we use the activity of metabolism and metabolic pathways as a druggable vulnerability in disease? Today, we are very lucky because we have available the powerful tools of systems biology, among which we have metabolomics, which will be the core topic of today's classes. But why metabolism? Like, why are we so interested in metabolites if we can measure genomes, RNA, like the transcriptomes, the proteome at different levels? So we do have other omics sciences that to some extent look more mature and have been developed earlier than metabolomics. Why? So, well, metabolites, if you look at where they are positioned in the system, they are really at the interface with the diet, the environment, and the microbiome. So not only they are a functional readout for the activity, so what is actually happening in the biological system, but also they provide an actionable layer, not only for easy measurements, for instance, in plasma, but also in tissues, but also to modulate those concentrations in order to affect the other omics layers. And this is true particularly because as many of you in this room are aware of, metabolites not only allosterically directly regulate the activity of proteins, but also they can affect their expression at the RNA level, acting on transcriptional factors. And also we start to appreciate that they also affect the expression of genes on the DNA. So really, together, this layer, it's able to affect the phenotype. And when we talk of phenotype, we can refer this word to different levels, right? We can go from the single cell to full systems, going through tissues, organs, and so on. And what is important, and we will go through in the next days of these classes, is that we are not only interested in understanding the absolute concentrations of metabolites, of their activity in a single cell or in the system, but it's really how the metabolic pathways are expressed in tissues, meaning over space across the heterogeneity of that tissue, that will enable eventually the function of a system, right? Like as we know, we all start from cells that have the same genetic background, but the expression of this genetic background into pathways that are functional in a differential way across the system is really what determines the function. So Said like this, it sounds simple. We just need to measure the metabolites, and then we will have tons of information on these layers and how to intervene. However, when I'm reporting in this slide the Kyoto Encyclopedia map of genes and genomes, and this is considered one of the most comprehensive maps of metabolic biotransformations across metabolites that we know in humans and other organisms. We tend to look at metabolism as a two-dimensional problem on a map with a lot of complexity, as you can see, but pretty much defined in our biochemistry books. However, the reality is more complex than this. The reality is that in the biological systems, so the reality is that this map is not defined yet. There are many, many metabolites that are detected in the signals when we see through mass spec and untargeted measurements. We see this signal being present. We are able of defining that those are metabolites. However, those metabolites have not been characterized yet, nor in terms of structure, chemical structure, not in terms of activity, of pharmacological activity and in our system. So this is a big problem, a problem that is bigger than we think, and particularly because it's not only two-dimensional, but this is really a network in multi-dimensions. So it looks really like a more multidimensional problem. In addition to that, this multidimensional problem is framed in the context of proteomics regulation, protein regulation, transcriptomics regulation. So it's really a problem that we can investigate only through bioinformatics modeling. So many open questions beyond like questions specific to disease, particularly how many unknowns and metabolic connections are present. This is still an open question, right? As students, we tend to think that most of the knowledge that we need is in our biochemistry book when it comes to metabolism. However, the reality is more discouraging than that. There is many, many unknowns, and the metabolic conversions are not yet completely defined. So if we flip the coin, it's an exciting opportunity for research to add to this field. But also their biological function, right? Like we started historically to think of metabolites as a means to provide energy and building blocks for the cell, to build the cells. However, these molecules are really regulating the system. So what do we do what do we use as an analytical toolbox for measuring metabolism? There are different approaches. In our research, we mostly use mass spectrometry, and I would say that today worldwide mass spectrometry is the approach that is mostly used. There is also NMR, nuclear magnetic resonance metabolomics approaches. That will not be a topic of these classes. There are advantages, very briefly, for those that are interested in the difference with NMR metabolomics. NMR-based metabolomics is usually less sensitive, significantly less sensitive than mass spectrometry-based metabolomics. But it has the advantage of identifying the metabolites through already in the raw data that are acquired from the samples. So the identification of the metabolites, it's faster because it's already incorporated in the original signal. While for mass spectrometric metabolomics, we will have way more sensitivity, way more broad coverage. However, the identification beyond the annotation of the metabolites is a more complex task, as we will see. So when we talk of mass spectrometry-based metabolomics, we have targeted metabolomics, which means we have a priori hypothesis of which pathways are changing in our experiment, or we are interested in studying, for instance, the TCA pathway. And then we set out a priori to set the parameters in our mass spec to only detect and focus those metabolites that are present in that pathway. So we will be blind to everything else, but we will be very, very accurate in identifying the metabolites for the pathway that we are interested in, and we will garner quantitative, not semi-quantitative, but quantitative measurements for that pathway. And this is an approach that is very useful and historically the common approach that that preceded metabolomics, right? That was, let's say, like classic analytical chemistry, analytical detection of metabolic pathways. But it comes with the caveat that it is really blind to anything else that can happen in that pathway. With global, also called untargeted metabolomics, the mass spec is set in a way that it can profile all the signals, the metabolic signals that can be detected in that specific sample. And what this means is that we garner information that is really agnostic to our initial hypothesis, but it is more collecting metabolic signals that are called metabolic features. And then through bioinformatics, the convoluting those signals that are significantly changing in the experimental conditions, and then following up those signals in order to understand to which pathways do they belong, what is their identity, and then building from that information. Any questions so far? Okay. So how does a global untargeted metabolomics workflow usually look like? We start from our biological model or the samples, right? It can be cell culture, an animal model, any type of biological sample. Of course, the sample preparation will be different depending on the specimen that we start from. Then the first step is collecting the untargeted high-resolution metabolomics profiles, which are collected by liquid chromatography connected to mass spectrometry. How many of you in the class have hands-on experience with LCMS? Okay, not a few. So once that we have profiled these samples, because of the nature of the chromatography or chromatographic separation in this approach, usually to garner a complete coverage of metabolome, we need to profile the sample under different conditions. For instance, using different chromatographies. And this is, I would say, is one of the biggest differences with transcriptomics and genomics, where you have one technique that has a certain depth of measurements across the genes that you're interested in. Here, depending on the nature of the molecules, meaning whether they are more hydrophilic or more lipophilic, we will have to complement the profiling through different approaches. Usually it's two different profiling and then complementing them and integrating in the metabolome and lipidome or similar. We will see this more in detail later. Next, there is data and pathway analysis. And then finally, because it's an untargeted workflow and it's not a targeted workflow, the metabolite identification happens at the end of this sequence, right? So at that point, we isolated the metabolic features that are significantly changing across experimental conditions. We go back to the mass spec. We set out something which is called a fragmentation experiment, MS2 experiment. At that point, the mass spec will isolate only the metabolic features that we are interested in, will fragment them, and those fragments will be characteristic and indicative of the identity of the metabolite. And then at that point, that will give us confidence on the identity of the molecule, which eventually is confirmed by comparison with standards. So going more in detail, let's say this can also be done at high throughput, right? Like it's not only, it's a very valuable technique for scaling up in a high throughput approach. The first step is chromatographic separation. The mass spectrometric detection, in this case, that instrument is a Q-TOF, but there are different mass spectrometric detectors that we will see later. This will generate the raw mass spectrometric data that look like that as a total ion current chromatogram. And from there, data processing, including peak peaking, retention time correction, and we will go through all of this later. This will bring us with a final list of metabolic future with their intensities, and then from there, we will perform statistical analysis, both univariate analysis, multivariate analysis, pathway enrichment, modeling, and systems level understanding. So now, any questions so far? No. Okay. So the first step in generating these metabolomics profiles is applying the chromatography. And the reason why we apply the chromatography is to some extent simple, but really important for the quality of the data. So any of you has experience with chromatography? Okay. Any idea of why do we use chromatography before the detection by mass spec? To separate? Yes, to separate. So, and that's, but why do we need to separate? So the reason why we use chromatography before the mass spectrometer is that you can imagine your sample as a complex mixture of analytes in the order of 1,000 and 1,000, right? Like 5,000, 10,000, like really a very, very complex mixture. If you would have to inject and analyze that mixture directly in the mass spec, in the first step, which is the ionization step, you will have all these analytes competing for ionization, meaning that some will suppress others. So the most abundant ones will suppress the ones that are low abundance. The ones that ionize easy will suppress the ones that are more difficult to ionize. So in order to increase the sensitivity and resolve your mixture in a way that you really gather understanding of the metabolites that you have in your sample, the chromatography helps to simplify what you finally inject in the mass spec at the moment of the injection. It's not only that, it's also that by the interaction of the analytes with the mobile phase and the stationary phase in the column, depending on the physicochemical nature of the molecule per se, those analytes, those metabolites will elute either earlier or later in your chromatogram. And that informs us. So the time is called the elution time, like the retention time, the time of the metabolites eluting out of that column is an additional dimension that informs you on the identity of that metabolite, directly related to whether that molecule is polar, is apolar, and how it behaves in terms of solubility between the stationary phase and the mobile phase. So that exchange is really what is determining this separation. So normally in the lab, in this step, you would have the mobile phases, and this elution happens in the gradient, meaning that you have two mobile phases. Usually one is more aqueous basis and the other one is anorganic solvent. And there are different combinations depending on specifically, like if you want to focus more on a part of the metabolism, or what happens in practice in the lab usually is that you have some very broad hypotheses. Like for instance, I do expect central carbon metabolism being of interest in my study, but I also want to see everything else that I can measure around that. So because central carbon metabolism is mostly made of polar metabolites, then those mobile phases and the gradient throughout the column and the column per se will be selected as to make sure that you have visibility, you have detection ability for those metabolites, and then also measure everything else that can be measured through that. So different options, we will see some later. There are some gold standards platforms, meaning LC and mass spectrometry as a connected system as a whole. That are mostly used for focusing on central carbon metabolism or lipid metabolism, or for instance, amino acid metabolism. But I would say there is usually in the community a consensus of using either certain type of mobile phases as opposed to others and certain columns, so that there is also data interoperability and the ability of making data meta-analysis across previous studies. So it's not as diverse that one could think from this slide.
Next question.
Yes.
So in this step, and we will then separate the metabolites, which is like protein binding or membrane binding, or either does it have any like tag or whatever you can distinguish where are these metabolites coming from, either from extracellular or intracellular. If you use a sample like plasma or tissue samples.
Yes, well, so I think this is two questions in one, right? If I understand it correctly. So one is what is the separation based on inside the column? And we will see that in the next slide. We will have a better idea of this. So let's leave this for this slide. The second question is how do you identify whether it's plasma, whether it's another type of tissue, right? So, well, I would expect you can realize, like when you do sample preparation, right? You know if it's plasma or it's other. Are you asking whether you can recognize the type of sample from the metabolomics data, or can you?
Yes, so I'm wondering if I just simply use plasma, so I know that most metabolites I will be detecting which in plasma system. But if I use the tissue samples, it might have extracellular.
Yeah, I see what you mean.
Within the tissue, but not outside the cell. Or majority they might be intracellular. So since this the sample going to be dissociated as a lysate, they might lose the information.
Yeah, you lose that information with this approach.
Whether there was a method to distinguish that source.
So classic global metabolomics is in bulk, right? So you lose that information because it doesn't have the resolution to go into like space resolution, right? But in the coming days, we will see another approach of metabolomics, which is called mass spectrometry imaging based metabolomics, where you do have resolution over space. And that is what usually people use when they want to see how metabolites are distributed between extracellular versus intracellular. However, the problem of extracellular versus intracellular within a tissue for metabolomic studies is still in development. And the reason is because it takes a combined level of resolution over space, but also sensitivity within that resolution. And these two aspects are connected. We will see that in the coming days a bit more in detail. But here, what we are doing is, this is in bulk analysis, right? So in practice, let's say that you have plasma samples, you will have a protocol that you develop for yourself where you have a certain amount of volume of plasma for each one of your samples. Let's say 100 microliters, right? And then that would be your, the measurements will be the average across that volume. And if it's a tissue, that would be, for instance, 5 milligram tissue. And the metabolites that you measure is the average across those 5 milligrams. So I would say with this technique, the only option that you have for understanding heterogeneity in tissues is already having, for instance, histology or an understanding of which parts of the tissue are relevant to you and sampling them differentially. But you will not have that resolution with bulk metabolomics. That requires like more single cell spatial approaches. And that's the frontier of metabolomics. So we will see it in the coming days. And we will also see how this approach together with that approach are usually used together to define different facets of the question, because this has sensitivity and better coverage, but that one has the spatial resolution, but lacks in sensitivity. It's very difficult for identifying eventually in MS2 experiments. So it's operationally in the lab, answering that question is more like a combination of techniques as opposed to just one. Yeah, okay. So your first question, like how does the separation happen in the column? So within the inside the column, it looks usually like this. We can have many different types of columns, but these are the ones that are mostly used in metabolomics. UPLC, so ultra performance, which means that the diameters of the silica particle inside the chromatography is in the order of the micrometers. And these silica particles are covered with the stationary phase, which we will see later. It can be a combination of C18, C8, amine phases in what is called reverse phase chromatography or hydrophilic chromatography. And you can imagine the mobile phase just flowing throughout those particles. And while the metabolites interact with the silica particles covered with the mobile phase, these metabolites interact. Depending on their nature, they stay longer or shorter attached to those particles, and that's the foundation of the separation through chromatography. So we will see literally in terms of retention time, some metabolites eluting out of the column earlier, some eluting out of the column later. And that's based on this interaction. Any question? Yes.
So the stationary phase, is it polar or not polar, right?
It depends. It depends, yes. We will see some later, like the reverse phase is apolar. The helic is polar. And you use them in different conditions, like meaning when you want to retain polar, then you usually use the polar one. So central carbon metabolism will be a helic column, right? When you want to measure things that are somewhere in between polar and apolar, and usually the most versatile one here is the reverse phase, which is more based on, so you can imagine these C8 or C18 hydrocarbon chains on the surface of the silica particles. And those are the chains that interact with the analytes, right? So different types of retention, different strength of retention, but also keep in mind that the composition of your mobile phase with those two solvents that we saw before, right? And the relative changes that happen over time while these metabolites are passing through the column, it's also determining the affinity of those molecules for that stationary phase, because that mobile phase changes composition over time, and it's called a gradient. And the user, like you, define the gradient. And the reason why you do so, because by modulating that, you can tweak how fast these analytes are eluting, right? Like, are they separated fast? So you can make adjustments and optimizations which are usually tied to what type of samples do you have, what type of molecules you want to separate, and considerations of that type. Okay, so now we started from this complex sample, very complex mixture. We went through the chromatography. It elutes out of the chromatography at specific retention times for each of these fractions. Separated. So we have a way less complex fraction at every moment that is separated and now can be injected in the detection measurement. That is the mass spectrometer. So the mass spectrometer is, there are different types of mass spectrometers, but you will find always these four components in it. One is the ionization source, then the mass analyzer and the detector, and then there is vacuum pumps, because all this system works under vacuum conditions. So the ionization source. So we have these molecules. They are separated, eluted. However, in order to be detected by the mass analyzer, they need to be charged. So we need to either add or remove one charge from these molecules. And this happens in the ionization source. There are different types of ionization. Gas phase, we will not go through gas phase because they are usually a more what is called hard ionization, meaning that while ionizing the analyte, they also fragment the analyte. And that's something that we don't want for, we want to avoid for untargeted approaches. And then there are the soft approaches, particularly the most common one is the electrospray ionization. It's the one that you will mostly find across metabolomic studies in repositories or from collaborators. It's just the most versatile source that is present on the market. It's usually operated either in the positive mode or in negative mode. And what that means is that in positive mode, it will add one charge, 1+1 on the metabolites. For some metabolites, it can be more of that, but usually, let's say the assumption is adding one charge. In the negative mode, it adds one electron, right? So you will see the molecular -1 in negative. In positive, you will see the molecular +1 as a result of this. So there are other approaches, the APCI, atmospheric pressure chemical ionization. This is using more a chemical soft ionization. You obtain similar type of molecular ion. Secondary ion mass spectrometry, many other types, MALDI and DESI for imaging. We will see MALDI in the coming days. And the reason why MALDI is used in imaging is because it has a laser that has spatial resolution. So if you imagine a laser that just hits your sample at different with different spatial resolutions, that's just more apt for that approach. So next, the mass analyzer. The mass analyzer, the most common ones are either the time of flight or the orbit trap. Any of you familiar with these names? The time of flight, yes, correct. Okay, so what is the difference? So the orbit trap is usually a high-end instrument, so like, way higher bar to acquire it for a lab, but it has way higher resolution in discriminating across molecules, across peaks, and the accuracy is way higher, meaning it has an error of usually less than 1 ppm. Mass range is very high, and so the time of flight is usually an average resolution. Still, time of flights are good for metabolomics. So usually, time of flights is considered good for metabolomics, but is not extremely performant when it comes to proteomics. The orbit trap is an instrument that is more versatile. Like you can push the measurements in proteomics, and at the same time, for other type of workflows, you can use it with a high accuracy and quality for metabolomics. But it's way more expensive. It's two different range of resources needed for this. In terms of detection, the detection principle, it's also slightly different, I would say. So for the time of flight, you can see the sources over there, the ESI source. Now you have in blue your ions. Those are ionized, either positive or negative, but have a charge. In the first part, there is a quadrupole. This is a quadrupole, and what is a quadrupole is I would say, is one of the first detectors that were invented for mass spectrometry. Today, in this type of high-resolution mass specs, it's mostly used for filtering out ions. So normally, in profiling metabolomics workflow, this quadrupole is open, like it just lets pass all the ions. But when we do MS-MES2 fragmentation, that quadrupole is the one that isolates, like that only focus on the ions that you want to fragment later. So, but it has a very low resolution. Then past that, there is this part which looks like a tube, and this is a time of flight. There is a difference of potential, and the ions, when they start there, they are separated based on what is called the mass over charge ratio, right? Like depending on how heavy they are and how big is the charge they have, then they interact with this difference of potential, and then they get detected at different times. And based on that difference in time of flight, the software is able to deconvolute the mass over charge ratio. So the readout here is mass over charge. Yes.
Basically, this slide about the separation. So basically, it sounds like you have different type of columns, and that fits for different type of the molecule.
Yeah.
So it sounds like once you select a specific column, you are specifically going to detect a certain subset of the metabolites, right? I see. So it's kind of a little bit biased, right?
Yeah, it is. Yeah, and that's the main difference of when we say global metabolomics, we would love to say that it's like doing genome profiling, right? That you just measure everything you can at that depth of measurement, or transcriptional profiling. The reality is that because by understanding how the detection is performed is highly based on the chemical nature of the molecule. And therefore, because the metabolism, like that pathway that we saw before, has areas that are lipophilic, areas that are more hydrophilic, like polar, right? What happens in the reality is that you will end up often using two different types of platform arrangements, like meaning, and also the polarities, like the positive, the negative polarity. You will have, for instance, the amine, they are seen very well in positive mode because they tend to get a proton easily. Other are less so in negative mode, right? So I think that's a learning that you gather both from literature and from classes like this, but also by getting your samples and running them and see what you annotate and then putting on pathways and see. So it's not to some extent is definitely broadening your perspective at the systems level, allowing you to detect changes that normally you would not think of in across your experimental conditions, but it needs complementary profiling. Yes.
Separation. You will have like like you will collect everything diluted from the column.
So you can imagine, actually, you don't get that fraction because of each fraction, like you have the sample, that sample flows, so it's fractionated at the end of the column. Then from there, it goes directly in the mass spec, and then it's consumed through the detection. So you don't really collect it at the end.
Okay, so basically just everything, most things from the column goes into the mass spec by their position within the column.
Yes, yeah.
So it's basically just
yeah, it's a full workflow. Like all of this in reality, you only see when you put the vial in the LC sampler, right? And then from there, you see the raw data at the end, and that's and the rest goes to the waste. So you don't really isolate anything. You can, if you want, if you disconnect the LC and you see where your metabolite of interest is eluting over time, you can, in theory, just disconnect the chromatography and collect fractions with a vial if you are interested, for instance, in testing that specific molecule. However, there is a caveat to that because, yes, the chromatography separates, but you want to be careful of understanding whether in that fraction you have only one molecule or you have, for instance, 5 different molecules that are all similar. For instance, the isomers, they tend to elute very close because they are usually very similar in structure and behavior, right? And that is an information that you gather from the mass spec. So in those type of situations, you would play with the gradient in a way that you can separate those isomers better and then try to separate.
Because I know it's for proteomics, people sometimes do fractionation. So it sounds like for the metabolomics, it's not the case.
No, it's not the case. It's also because the sensitivity for this type of mass specs is usually good. So you don't need to pre-concentrate in this type of approaches. Yeah.
Profile both lipophilic and hydrophilic and hydrophobic both columns?
Yeah, so usually the helic column, because the stationary phase is polar, you will tend to use that more for polar compounds, while the reverse phase, which is the hydrophobic, the C18, the C8, those are mostly used for mildly polar or non-polar fractions. But the C18, you will see, is the most common column used in analytical labs. And you can separate things that are slightly polar or so. It's just, I would say, the strength of retention, right? It depends on your mixture, like how many of the lipids do you have, how strongly they are retained in the column. It's more really, I would say, an experimental learning. When you have your sample, then you can learn from the sample how it behaves across the column. So these are more like the general concept and how you would operate decisions based on what you're interested in. But then within those decisions, you still need initially to go to your mass spec, start profiling your quality control, like as a pool of the samples that you want to measure, and see how is the signal. Do you detect the metabolites that you're primarily interested in? Do you see much other signal around? Is that lipids? Is that more polar things? How does that project on the pathways? And then from there, there is usually a point where you're like, okay, this method is good enough for profiling what I'm interested in at this point, right? And then you go ahead from there. But it's really, and this is one of the reasons why metabolomics has developed across omics sciences as the younger one, because it has more complexity connected with the detection.
In abnormal situation in UCLA, the other parameter metabolic, do they do both hydrophilic and hydrophobic, or
it depends. So for instance, when you say lipids, right, like there is some classes that have mild polarity, some that are completely hydrophobic, right? So the ones that have mild polarity, you will still see them in sometimes with the central carbon metabolism. The ones that are completely hydrophobic, you usually don't see them there. But also you want to consider the sample extraction step, because when you extract the sample, that's also where the bias happens. So usually when you are more focused on central carbon metabolism, you will use solvents there that implicitly will leave behind the most apolar lipids, right? But if you already know that you are interested in the apolar lipids, then you will already, from the the sample extraction, use different solvents like tert-butyl methylator or chloroform, working in glass. So yes, like these are all considerations that while it allows you to look at things that not necessarily only the ones that you hypothesize, right? So it's very powerful because you can measure 5,000, 10,000, even more than that metabolic features at a time. However, it's still it will still give you a partial coverage, and there is bias. Okay, so this is the time of flight, the orbit trap. The orbit trap is way advanced approach. The detection here always happens based on mass over charge. So what we measure is mass over charge and abundance at the end. But the separation happens through letting these ions orbit around that fuse. And what happens here is that between the walls and the fuse in the detector, there is a difference of potential. And the frequency at which they orbit around that fuse is, there is a Fourier transform that happens there, right, to deconvolute that signal. So it's a way more complex signal. It's not as mathematically easy to deconvolute that as a QTOF, as a TOF. But fortunately, mass specs and they already do it. It's a deconvolution that happens within the software, so that's something that we usually don't even see. But it's based on a Fourier transform of that signal. The overall result, like the part that interests you of this, is that it has way higher resolution, better accuracy. And therefore, if you were to do proteomics, and you knew, like my lab wants to do proteomics and some metabolomics, you definitely would want to go for an orbit trap. But if you would be like, I only want to do metabolomics and targeted in my lab, and I have to allocate resources correctly, then the TOF, it's an instrument that is good enough to do so. Okay, so it's 10. I would call a break, and then we continue after 15 minutes. Okay. Okay, so now we have seen liquid chromatography, mass spectrometry. We have profiled the samples. How does the signal look like? So this is the overall total signal that comes out of the LCMS analysis. It's called a chromatogram. You have the time, the retention time, and this is every second, like every moment when the fraction from the LC has been injected in the mass spec, right? Like you can see this as slices. We will see it in 3D after this. So, and then the abundance, the measured abundance. In this case, this is the sum, the total ion chromatogram, meaning that these abundances are the sum of every ion that has been measured at this specific retention time. So you will have low affinity to stationary phase of the column. So molecules, metabolites that are low affinity to the stationary phase, they elute very fast, right? It's intuitive. They don't stay in the column long, they just elute out. And the high affinity to the stationary phase, you will see them later on in time because they take longer. They like the stationary phase better, so they stay longer in the column and they elute later. The same chromatogram can be seen in 3D because it's a way more complex information-rich piece of data. So before, we were looking at the chromatogram from here, from this dimension, right? Intensity, abundance, and retention time. But what we did in the former slide, all the signal that is in the M/C dimension, we just squeezed it here and we summed it up, right, in the total ion chromatogram. But for each second, for each retention time moment, you can also slice out that moment and see all the M/C values that have been measured at that specific moment injected in the mass spec, right? So meaning that, yes, we inject fractions, but we say those fractions may have more than one metabolite, right? It's rare that it's only one. So if you want to see what was injected in the mass spec at that specific time, then you would select the time and just see the slice, for instance, the yellow one, and that will show you the M/C values, the molecules that were there at that time. Any question? I know this takes a little time to orient in. Okay. So based on this output, let's define what is a metabolic future. A metabolic future, at this time we can talk not of metabolites, but of metabolic futures. Metabolic futures are those signals that are, one, consistent in terms of shape and pattern with what in silico expected for metabolites. So we do have an expectation of how those signals would look like for real molecules, for real metabolites, right? So a metabolic feature is something that has that is consistent, is measured consistently with that type of pattern and shape of peak and intensity, and is defined by an M/C value of the molecular ion. Therefore, like, let's see, if we slice through here, that would be like, for instance, this one for this molecule, this one for this molecule, and so on. Our retention time, right? So for this one would be here. And then these futures can come in what is called adducts, in different adducts, meaning that the same molecule, the same metabolite, can be corresponding to more than one signal there. And the reason for that is because in the mobile phase, but also in the biological fluids, we have we have sodium, we have potassium, there is a lot of water. So these molecules can create an adduct with sodium. Some are more keen to do that, so some are less keen to do so. But what happens at the end is that often for some molecules, we would see an adduct with water instead of the molecular ion, and then you would identify it by fragmenting and deconvoluting that signal, or an adduct with sodium or an adduct with potassium. Now, you would say, how do I know, if I don't know the identity of the molecule, how do I know whether it's a molecular, it's a sodium adduct, it's a potassium adduct, and so the bioinformatics helps you deconvoluting that signal because it gives you all the possible options for a specific M/C value that is calculated, and that's one of the reasons why the resolution and the accuracy is so important on the mass spec that you use, because the higher resolution, the less error that you can use for annotating and inferring computationally the possible molecular ions and adducts that correspond to that signal. But also over time and with literature and with experience, you will see that there are patterns that you can learn there. So there are, for instance, some molecules like glucose. Glucose, you can measure it as a sodium adduct, right? Like it's almost impossible to see it as a molecular. But it really depends. Another aspect is that while the adduct will have a mass over charge different than the molecular, right? Because it has either sodium or potassium or whatever is attached to that. The molecular, if measured, will be measured exactly in the same retention time slice. So that's another indicator that is used by the bioinformatics software to decide whether this is likely an adduct or is a completely different molecule. Because if it's if it has a mass difference that corresponds to the common adducts, and the molecular is also in the exact same slice in retention time dimension, then it's very likely to be an adduct of the molecular. That makes sense, right? It's the same molecule. It came out of the LC at the same time. It's just present either as a molecular or as an adduct, but it's the same molecule. And that's how multiple futures can stem from the same molecule. Yes.
So I get how you're getting retention time and mass to charge, but I'm just curious what that intensity now brings into it as well.
Intensity is the abundance, like how much of that molecule is in
So
the...
that's just based on like to get relative quantification, right?
That's a good question. So now, if you were in targeted, right? If you were in targeted, meaning you would target specific molecule, you would, in that case, you would have designed this analysis as to have also internal standards. And in that case, you would be able to do quantitative, not relative, because you would have standard peaks in it. And for those standard peaks, you would know that abundance to how much it corresponds in absolute quantitative terms. And therefore, you can calculate your signal. But because we are in untargeted here, we don't have usually internal standards. Sometimes you can add them, but in general, the quantification is called the semi-quantification. And also the variability of the signal around the same quantity for the same metabolite in untargeted is higher than in targeted. In targeted, usually you would say a method is reproducible if you are around 5% error, right? Like the variability, because it's supposed to be optimized for measuring that with the standard. In untargeted, that can go up to 30%, right? Meaning that in untargeted data sets, that's a very good question, actually. In untargeted data sets, in order to say that something is changing, you need to set a fold change that it's significantly accounting for this expected variability. Does it make sense?
So if there's like an extra dimension that would be necessary to get finer tuned data, what would that be?
Well, the internal...
Everything plays its own little part.
Yeah.
Right, like intensity, abundance, retention. It's going to basically show you kind of what the molecule looks like. Mass to charge is going to give you a little bit about, the atoms that are in that, itself. So, like, it just kind of like speculated the future. Like, what would be like would an extra dimension be helpful?
Oh, yeah. There are indeed instruments that have that extra dimension, right? Like, well, beyond the imaging that we mentioned before, but that's a spatial dimension. There is there are instruments that have ion mobility, it's called, where basically you also have a counter current that the ions face while in the detector, and that is helpful for discerning isomers that have the same mass, meaning same mass over charge ratio. They look very similar, so like usually here they will look almost the same. However, because they are isomeric in the space, they are different, right? Like three-dimensionally, they are different. So if you throw at them a current of literally like it's a gas that is thrown like with a certain velocity, they will behave as a resistance differently, and therefore will hit the detector differently, and it's called ion mobility.
But do you typically do that post hoc? Like,
You don't that's it's just like an additional dimension of complexity in the data. You can do that if you have a reason to believe that it's important for you to discriminate those isomers. Like usually that's the logic behind. Yeah. Yeah, and then the other dimension is the standards. Like for instance, what some methods do, they a mix of standards that of course cannot cover all the molecules that you have in untargeted because it's thousands, but they cover one standard for each part of the either an visit value or the retention time part, so that you can see how these signals behave at different windows of retention time. And that gives you a bit of like an improvement of your semi-quantification, but it's always semi, right? Like this is untargeted. The normal workflow would be, this is discovery, right? You look which pathways are changing that you weren't thinking of. Once that you have those hypotheses and you're pretty confident of which molecules are they, then you would still switch to targeted and go more highly sensitive, able to identify in the first run, right? So, yeah. Yes.
So I just want to confirm, so basically, with untargeted metabolomics is semi-quantitative, it cannot be quantitative.
No.
We only have kind of just internally relative.
Yeah, like if you would see, I don't know, like some data that say we did untargeted metabolomics and we found a 5% difference in these two metabolites, in two different conditions, and it's untargeted metabolomics, I would be very, because it's not quantitative. It doesn't have an internal standard. Of course, then, every case is different, but that's the general rule. Like that's, there is more variability because you cannot focus on optimizing the detection of everything while you detect everything, right? That's targeted. So it serves different purposes. Okay, all right. So now, stationary phase. This is what we saw before. So as we mentioned, yes, it's untargeted, but it doesn't have coverage on everything. And so the two main options that are usually used is for hydrophobic lipid metabolites, C18, hydrophilic, aminopropyl tetracarbon metabolites. And these two approaches, like these two combinations, which are called in turn helic and reverse phase liquid chromatography, they tend to give like together. There are some publications that show that if you run these two different profilings, you have a high likelihood of having captured the majority of the metabolism that is currently relevant to our studies. Of course, it's an approximation, but if you want to be in a place where you have good coverage on the metabolome, lipidome, and so I would suggest these two, and then also adding a C18 run with an extraction method that allows you to focus on the really apolar extraction of lipids. Okay, so just summarizing everything we saw, there are many, basically 3 pillars that we need for doing this analysis. Analytical chemistry, LC, mass spectrometry, we saw that. The bioinformatics part, we will see it now. And then the spectral databases, right? Like there is not that much work that we can do here, but we can know which databases are the most comprehensive ones, how to use them, and how to implement it in the bioinformatic workflow so that we can have an annotation of this list of molecules that we will see later. It's not an identification, but it's an annotation. Okay, any questions so far? So molecular databases, there are many databases. Some are open source and others are less so, like some have limits of usage, like Kathleen has some limits of usage. The other ones are open source. I believe this number has been updated to higher for GMPS, but in general, you have different options depending on the resources that your lab has, and so you can choose what makes more sense for your studies. if you're focused mostly on things that are known, like metabolic pathways that are known, they all contain that. Some have more drugs and xenobiotics, others have less so. If you are doing exposomic studies, meaning you're studying the exposure to toxicants, and so you may want to use specific databases that have more are more populated towards that. So what is a known metabolite in terms of identification? So there are different levels, and it's also, we can also look at this as like different level of confidence in the identification. So the first one is not identification. We cannot call it identification. It's putative annotation, where we take the M/C value measured by the mass spec and we match it computationally against a database of known molecular identities with the relevant M/C value, setting a specific error in ppm. And that error depends on your mass spec on the platform, right? So again, that's the interesting part there is that how good of a mass spec you need for, because what you want to consider is that after a certain threshold, you will have molecular metabolites that are isomers, meaning they have the exact same mass, right? So having a better accuracy on the detected mass, sometimes for metabolites, it doesn't really solve your problem of discriminating across isomers because the mass is exactly the same against the databases, right? While this is very different in proteomics because you have multi-charged molecules, peptides that you are detecting there, and to discriminate those multi-charged patterns of peaks, that's the resolution is really important there. But for metabolomics, as I said before, a QTOF, a good QTOF makes the job. Yes. No? Okay. Then in-source fragmentation. So the second layer, like you have this molecule, the mass that you measure is matching the mass that you have on the database with 5 ppm error. Fantastic. It usually gives you like 5 to 10 to 20 different molecules that match that value. How do you decide which molecule it is? So one, you can look at in-source fragments, meaning that in the initial electrochemical ionization of the molecule, it can happen that the molecule fragments partially. So if you slice out the retention time for that molecule and you see for fragments that are typical of a specific metabolite, that's something that makes you more confident that that is the identification as opposed to the other, but it's not yet at the level of identification. What you normally do is that you will have a list of molecules that are interesting to you. You will set up a fragmentation experiment, meaning you will go back to the mass spec, and you will set up a method that at this time is targeted for fragmenting those unknowns. So with the M/C value, you will set the mass spec of isolate these M/C values at these retention times. The first part, the quadruple of the mass spec, will isolate them. Then there is a collision chamber where you set the collision energy to literally fragment, to break the molecules that you isolated. And then the high resolution part of the mass spec at this point is set for measuring the fragments. So the M/C value of the fragments, right? It's like I like this comparison because I remember when I was a student, it really helped me understanding why we do so. So let's imagine that you have two different glasses with different shapes, right? Like, and you weight them, they weigh the same. How do you decide which glass is which shape? Like based on the weight, M/C value, they look the same. You cannot, right? Now you take those glasses and you break them. They have different shape. They will get different fragments. And while you look at the fragments and you weight those fragments, because they break specifically depending on the structure, on the shape of the glass that it had, then you will be able to say, oh, because these fragments have this specific weight, this is glass A and this is glass B, right? And it's the same thing that we do here with the molecules. Like they weight the same, but they are not the same structurally. So how do we discriminate them? We take them back to the mass spec, we break them, we measure the fragments, we weight the fragments, And then we say, all right, these fragments are typical of molecule A, these fragments are typical of molecule B. Yes.
What's your fidelity? Like, is that pretty consistent, like when you break a molecule?
It depends. Like there is something which is called the diagnostic ions, right? That the number of diagnostic ions, meaning the minimum number and the weight of those fragments per molecule that allow you to almost unequivocally say this is molecule A. It changes depending on molecules. For instance, if you look into lipids have high modularity. And they have consistency in how they fragment across classes. But then when you want to, dive deeper into which specific lipid is this, right? Where is the double bond? How are the chains distributed across different ramifications? Then it gets challenging. But for small molecules, for some of the small molecules, you have very, very clear and definitive MS2 patterns. So it would be nice to have one answer to that too, but and I would say today there are softwares, we will see them in the next days, that allow you computationally to take a batch of MS2 spectra and match them across databases and see which ones are the best matches and scoring them. However, even in that case, there is a lot that has to do with the user training and really, the ability of looking into that mass spectra and validating what the software tells you it is. And finally, for publication reasons, there are different layers. Like it depends whether you have orthogonal ways to confirm that change in the pathway. Like, for instance, let's say you have transcriptional data, or let's say you have enough changes in a specific pathway that gives you the significance of change for that pathway that allows you to spend less time on going to using standards and identifying each single metabolite, because you can say, okay, this is likely the pathway that is changing. I have 10 m/z values that change in this pathway. So the likelihood of this being predicted as the dysregulated one is very high.
So context is important.
Yes, context is very important, and it allows you to save a lot of time. I've seen personally in my career projects, I wasn't involved in them, but I've seen literally people spending 2 years only on one molecule and that MS2 spectra to try to identify that unknown, because of interesting biochemical activity. So some of the most active metabolites were discovered like that, but it still takes a lot of time to move from an unknown MS2 to the structure of the molecule. And you need organic chemistry for the unknowns, because you will have to synthesize the standard and you will have to run that standard through the mass spec, and only that point will tell you if it's the molecule or not. So that is called the fourth level of identification, and that's considered the highest level, when you not only have putative annotation, you have in-source fragments, you may not have a consistency, but that's not a requirement. But the MS2 fragmentation pattern is a requirement, and then the comparison with the standards. Now, do you always need to go to that level for publishing your data? As I mentioned, it really depends, right? It's context dependent.
Yeah, the reason I'm asking is because, when they obviously they do untargeted metabolomics, and let's say you have a really, a metabolite that's just called X, whatever, 5-digit number. And they give you a lot of information in the supplemental table, right? And you're like, oh, well, this, metabolite has all these different metrics to it. That doesn't seem like that's enough data to help you identify exactly what metabolite is there, though.
So here is the thing, right? You can see data that are published at different. You can see publications that contains all layers, all of these levels of just the annotation. Some publication even just give the amino acid values, right? So as far as the publication, the data, the results, to not claim to have an identification at that level, it's fine, because this data is available out. the limitation there is that of limited interpretability, because you can do like meta-analysis sometimes, you can infer, you can predict things, but still you will have to confirm that through orthogonal confirmation or replicating that experiment and going through all the steps, right? So it's in that case, it's the publication per se will be of limited interpretability, but as long as you don't claim that you have identified something based on like a putative match, or as long as you're very clear in reporting the data of how you assign that annotation at which level of this, then it's robust in terms of fairness of having it communicated. But how enabling it is for like the next researcher to go and use that information for replicating and build on that finding, that's a different story. Yeah, but we will see later today or next time, depending on time, that there are nowadays it's always more requested to have the raw data uploaded to repositories, and in those cases you can go get those data and reanalyze them on so you can see that signal, right? And
So something like Metabolon, right? They would have probably uploaded their
oh,
unknown metabolites.
well, I don't know. Metabolon, I think, is a private company, right?
Yeah.
So I would refer to I don't know if they upload the data to a repository or not. So that's but for other studies that are done like in universities or so, often they do upload those raw data, or they make them. You can email the study lead and see whether they want to share that. But there are increasing policies for studies that have been funded with public funding of having those data released, and mostly you can find them in repositories. We will see later which repositories are they. So unknown metabolites, right? What do you do if you see that it's a highly significant change? It's likely a molecule from the signal pattern, but it doesn't match anything in the databases, right? So is that a metabolite? Is that not? And should you pursue that? So we will see that later. There is usually you go directly to fragmentation and you start from there. And there are databases that allow you to perform similarity search across fragmentation trees with molecules that are known, and that allows you to get some aid and help in formulating hypotheses. Also, if you're able to synthesize that metabolites, you can run NMR, you can run cryo-EM. Now there are some ways to use cryo-EM with small molecules that allow you to actually see the structure. It's very expensive, but it's possible. Synthesis and analysis of your compound, right? Like these are all upon having the compound available. Lipid maps is another database. It's very specific to lipids. So it has both lipids that have been detected and measured in biological systems. It also has lipids that are computationally inferred based on the modularity that we know of expected lipid classes. So these are where you can find most databases: HMDB, the Human Metabolome Database, Metlin, GNPS. GNPS is now having also MS2 data, a very good amount of MS2 data that allows to do fragmentation trees, and so it's a very good resource. The NIST dataset, MONA, MZ, Cloud. So I'm sure there are new ones that I haven't reported here. So it's evolving. This scene is evolving. Metabolomics data repositories. We have the biggest one, Metabolomics Workbench, is based in San Diego, and I would say it's the point of reference for North America. Metabolites is an initiative from Europe. Of course, it's accessible from everywhere in the world. And then there is a Metabolome Exchange that is a data aggregator that allows you to search for metabolomics archival raw data and studies across these two repositories and others. So this is where you would go if you would want to see whether former investigators have already studied something similar than you're interested in, and you're interested in downloading the data and perform reanalysis, meta-analysis, doing different grouping and answering different questions. Okay, so we are 10:47. So we are behind. So let's go ahead in the MS processing tools. So now we understood how we get the data, what the data look like, and pretty much a bit how to think around both the detection and the interpretation of the data. What are the bottlenecks? What are the difficulties there? So fortunately, we have tools, bioinformatics. We don't have to do that manually. Part of it is still manual, but most of it is, especially in untargeted, is through bioinformatics. There are these three main pipelines and tools that can be used for untargeted data processing: XCMS, Metaboanalyst. They all have also our packages, and MZ-Mine. Today, if we have time, or on day 2, we will see MZ-Mine and XCMS. But the tools are always evolving. This is one of the last comprehensive reviews. There are others more recent. So feel free to look into new things. But for detecting the signal so far, I would say these are the frameworks that I usually apply. So Exi. Mm-hmm.
So, what is the difference? So you are choosing MC, XCMS, and UZ.
Yeah, the differences are really, I would say, in I think this is also context specific. So we will go through in the next slides, explaining a little how they differ. But in reality is that they serve the same purpose of providing you the detection of the list of amino values, retention time, so the metabolic future, and their abundance, right? And then some of them allow you to do annotation directly within that environment. Some allow you to also match additional MS2 data and having another layer of information. Some are connected to Sirius, which is a software that guides you into the interpretation of the fragmentation tree, and therefore making hypotheses on the structure. Some are better suited for implementation in R when you have your workflow. And let's say sometimes you have different type of analysis, and the metabolomics that are just part of your analysis. You want something that runs robustly in R and it's easy to implement there. So it's really context specific, the choice. It's also, some have a graphical interface, a website. It's easy to use, like those that are not very familiar with R, and so they tend to, there is no need of learning R for running one analysis, but others that are willing to have things that can get embedded in R better, then they use other things. So they all have a bit of like pros and cons, but fundamentally they serve the same purpose. Yeah. Me, myself, I mostly use these three, and, depending on the study or the type of visualization that I like better, right? Or like, so, you would use interchangeably one or the other, depending on what are you doing in that specific study.
And which one is the top one?
Mm-hmm.
Which one is top level or advanced?
Oh, I would say they are all advanced. Like I don't think there is a unique answer to that. No, even the ones that, like for instance, Metaboanalyst has a very developed website interface, but also our package is there. It requires a bit more R advanced skills for being used in R Metaboanalyst as opposed to the R package for XCMS. But at the end, in terms of like processing the data, I don't think there is one that performs better than another. No, it's just different environments, and don't be. Also, sometimes there is a bit of the expectation that if you have a website graphical interface, then it might be less advanced than things that run in R. It doesn't hold true for metabolomics because most of these options, both in R, both in the software version for MZMine or the graphical web interface, they all allow you to set the parameters that are the ones that optimize your detection of the metabolic features. So no, I don't think there is an answer for that. It's more you have different options, and you will see there is differences in the data visualization that they offer, right? What type of plots, and even though most of the time you end up exporting the matrix of data and doing your own visualizations, right? But for some studies, you may prefer one or over the other because of that. Or like the Metaboanalyst one has also modules that allow you to do biomarker analysis, where you can have ROC curves and combined ROC curves, or like ratios of metabolites used as biomarkers instead of the absolute value of the biomarker. And Metaboanalyst also has a super vector machine and more machine learning embedded to that if you are willing to explore this type of modeling of the data. And that is directly plugged in the workflow, right? So you don't have to take your data out. And but can you do the same with the others? Yes, you can, right? That's in terms of quality, I don't think there is one that is superior than the other. It's more convenience and how the workflow is accessible either online or through R. Yeah. Okay, and we will see it here, right? Like which briefly describes what these packages do. XCMS. So we have this data three-dimensional. We have seen them before. The first step is converting them in a format that is accepted by most of these platforms. It's usually the MZXL format, MZXML format. Yeah, and this happens with a tool that is called ProteoWizard. There are other tools as well, but usually that's an easy choice. From the moment when they are converted, then we feed them into the pipeline. So the first step is filter and identify peaks, right? The peak picking part, meaning that the algorithm knows that how mathematically a peak looks like what is a molecule, like it has a Gaussian shape, it has a specific level of symmetry, and has an isotopic pattern that is defined in a specific way. And after it peaks, like it detects every signal that is consistent with the metabolic peak, it matches peaks across samples. So let's say that you have this batch of conditions in your analysis, then all those peaks based on M over Z and retention time, the software looks for those peaks in every other sample. And what happens, this is iterative, meaning that it does it once and then it goes back and double checks that it hasn't missed peaks in that retention time area. Then, which is filling in missing peaks. Then it statistically analyzes the result, right, like between control and experimental condition, and visualize important peaks. So this is simplified, but that's pretty much what happens. There are advanced settings that one can add, but that's the core of it. This is how the output looks like from XCMS Online. So you will have a long list of peaks, future ID, the fold change is calculated across condition, the p-value, whether it's up or down regulated, the amyloidosis value, the retention time, in this case is in minutes, the max intensity measured across all the samples, the average value for dataset 1, for dataset 2, whether there are isotopes, and the adducts that I mentioned before. So it goes through every slide of retention time at the same, so for this retention time, right? It would go and look whether is there any other molecule detected that is consistent with the molecular weight of an adduct for this one, right? So plus water, plus sodium, minus potassium. And you can set which adducts you expect, or you can just leave, the standard settings. And that's pretty much it. P group, we create groups depending on where they are. Yes?
So this won't really help you find an unknown metabolite, it just like tells you what you know or what it knows you have?
No, this is all the molecules, all the molecular features, which molecular feature being the M over C and the retention time, like each row is a molecular feature, right? And each row is a molecular feature that is potentially a metabolite, meaning that computationally that signal is consistent with a metabolite. But now, do you know the identity at this stage? No, you just have this list of metabolic feature in your samples. In this case, you also have the statistics on whether they change across the two conditions. But this can at this point, at this stage, they can have known metabolites, unknown metabolites. In the next step, the next step will be the step where you focus on identifying them. Right? Because that's happens, like you will take that M-Z value, match across the database. Is there any known metabolite that matches this value? If yes, then let's see if it's that metabolite, MS2, the fragment fish. No, there is nothing like that. So at that point, you go back to the mass spectra, and what you do is trying to understand, is this an actual peak? Is this an actual molecule? Because of course, as every software that runs massively across an information-rich raw data that has variability across the data per se, like in the one single raw data, there is variability. So sometimes some of these detections are artifacts. So before embarking, you cannot assume at this point everything is an actual molecule. These are molecular features, so take it as it is. It's something that is likely a molecule, computationally looks a molecule. It's detected by the algorithm with the parameters that we set as a molecule, but it needs further investigation. So Metaboanalyst, this XCMS was like that. Metaboanalyst has a bit more options, but it's very similar what it does. Data input, what happens, it accepts different inputs. You can have either the spectra, which is what we saw with XCMS, right? Then parameter optimization and peak detection, which is what also XCMS does. Peak alignment across samples, which is what it does as well. Integrity check, data filtering, missing values, normalization. This is also something that is done by AI. So this part is really the core that is usually resonates and is redundant across different workflows. Now, Metaboanalyst has a bit more expanded options, particularly the data input. You can also search just for concentrations, that a list of metabolites in concentration that you already have. Spectral beans or table, peak table, meaning you extracted that list that I showed you before, and you may want to analyze it through Metaboanalyst. So you can also start directly from there, or you can use also NMR here. Right? We will not focus on that, but if you have it, this software has that ability. Statistical analysis has not only the core of statistical analysis, biomarker discovery, time series, so way more options for statistical analysis. I mentioned before, output as well, like many different visualization steps. You can always export the data and do your own analysis in R or any software that you like. But I would say for those users that do not they are not very comfortable in R, yet they want to explore the data as much as they can visually, but also in terms of statistics, Metaboanalyst is a very good option because it's on a website. It's very easy to use. Okay, this is MZMine, which is the one that I would like to try with you in the hands-on experiment today. So MZMine has a big expanded capabilities. It's very modular in the way you can add and remove analysis steps in your analysis pipeline, both in the data pre-treatment, but also in the analysis in statistical analysis. So in blue, you can see the parts of the workflow that are for LC-MS and LC ion mobility MS or ILC-MS, right? So with MZMine, you can do either analysis of LC-MS data, but you can also do analysis of LC-MS data, and that in part possible also by XCMS and meta-analysis. And interestingly, it also has ion mobility in the data processing ability. So if you have ion mobility data, this is one of the softwares that can be very helpful for you. It also has a downstream very easy integration with Sirius and GNPS. And so why is this important? This is important because these are really helpful when you have unknown metabolites and you're trying to, make your mind around that MS2 spectra. What are the possible structures? What are the similar structures that are known across the tree of similarities for MS2? And so I know it's a lot of information, but I would say the take-home message is that MSAT mine, I tend to find it very, very useful when you want either expand towards ion mobility. It also has modules for MALDI and mass imaging. So it also has that aspect that has ionization, visualizations abilities to see the spectra in 3D. It's really, really flexible, but also the interesting part is that when you're dealing with unknowns, it's very easy to connect it to other options and tools that allow you to explore those unknown signals. Okay, this we already discussed, metabolite identification. We have an example here. So this is an MS2 spectra. MS2, electrospray ionization positive. You see many peaks, different. These are all fragments for the molecule. So how this is the precursor. So the molecular ion in positive mode is has an M/C value of 245.23. But how do we identify who this molecule is? We went already back to the mass spec. We fragmented it. We collected MS2 spectra, and you can see here that now 245.23 is not the dominant peak anymore because we fragmented it, right? So it's not, the molecule is not intact anymore, but then you see emerging these peaks. And these peaks are the fragments. So for instance, in this case, we use those fragments, 72, 100, 174. We go to Maclean, the database Maclean. We perform a fragment similarity search. We put some tolerance depending on the mass spec and the variability that we expect. And that allows us to start understanding the structure of the fragments. So it matches to this, which eventually points us to confirming that this is acetylamino-propyl spermidine. Okay, any question? Okay, so now we went through getting the metabolic features, the abundance, the different bioinformatics workflows to do so. What do we do once that we have a list of metabolic features, M/C values, or metabolite identities? These are significantly changing in the conditions that we are interested in studying. However, we are at the metabolic lab, we have our metabolomics data sitting in our lab, so one thing that we can do is by using repositories or even just data that our collaborators share with us, doing meta-analysis and looking whether the signals that we see are also consistent in independent studies, right? But on top of that, what we do at that point, we do pathway analysis, right? We take those data, we superimpose them to metabolic pathways, we see them in context, and we start to understand whether there is a significant enrichment of specific pathways that corresponds to the variation that we have in our data. And in addition to that, we can integrate that with the proteomics data and transcriptomics data to understand if that metabolic changes are corresponding to transcriptional regulations, whether there are changes in protein abundance, or it's something that happens only at the level of the kinetic of the enzyme. And for that, we have different tools. Particularly, this is for pathway enrichment analysis. So we have IPAT3, MetaboAnalyst again, Reactome, PathView. So many different options. We will see and try some of these in the next days. I would like to focus on one, which is Mamichog, the pathway prediction tool. This is a pathway prediction tool. So we pretty much have two options to predict pathways that are changing based on these signals. One, we go through all the workflow. We find the metabolic features that are significantly changing. We identify them. We go through all the identification steps. Now we have the metabolites, not the metabolic feature, the metabolites that are changing with their fold change and significance. And then we enrich pathways. But that takes a lot of time, right? That takes a lot of time. And once, by the time we go through all that work and resources, we might discover that we are not even interested in those pathways. So that's a problem, right? How do we bypass that? There is this tool which is called Mamichog for pathway prediction. So Mamichog takes the metabolic features, the hemoversity values and retention times that we saw before, and it calculates, so it matches them across databases of metabolites. And it just takes every annotation possible with that specific, with a specific error associated to that, depending on the mass spec, and says, okay, let's say that everything that matches these metabolic features is actually present in my sample. What if all these molecules would enrich the pathways in the metabolome? Like, which pathways would be the likely ones that consistently have changes based on these metabolic features? And at that point, it calculates a Fisher t-test of significance within pathway population. And that allows you to say, so the ultimate output that you gather for this analysis is starting from a list of metabolic features. You get a list of metabolic pathways that are predicted as enriched based on those features. And the strength of prediction is associated with the significance of the enrichment within the pathway. So meaning as more metabolic features fit a specific pathway consistently, the higher the likelihood that that pathway is actually the one that is enriched, right? And so the bottom line here is that if you run this before going through identification and everything, you will be able to answer the question, is there any pathway here that is of interest to me in this study? And then from there you can say, okay, well, but this is just annotation. It's I feel confident this is something that is informative for my project. Therefore, I'm going to pursue it through putting more resources in the identification. Okay, and then as we mentioned prior to that, after that we have different layers, transcriptional protein layers. We can use PathView, Reactome, XCMS, MetaboAnalyst. They all have the ability of integrating this type of data within the metabolic pathways. Let's take a break. 15 minutes break, and we will restart at 11:25. Okay, so last we will look a bit into the workflow of how do we approach an unknown metabolite. So usually these appear like metabolic features. So the bioinformatics, the signal, our own manual analysis and validation of that molecule signal is checked green for, okay, this looks as a molecule. This looks like as it's a metabolite, right? So, but we try to match it to the repositories of known metabolites and the literature as well. And we can't find anything that matches this molecule. Then we need to de-risk. It's not an adduct. It's not an in-source fragment. It's not a contaminant that is made, like, for instance, dimeric forms of other molecules or so. Then, well, that's likely an unknown molecule. How do we approach that? So we need to start from one, the accurate hemoresidue isotopic pattern and retention time. So from there, once that we have the accurate hemoresidue value that we garner from a high resolution accurate mass spectrometer, the isotopic pattern, we garner it from the mass spectra, and the retention time from the chromatography. Then using this information, we can use computational tool to calculate molecular formulas, like possible molecular formulas, meaning that given this value of hemoresidue value measured, what are the, and this ppm error on the mass that I expect, how many different combinations of molecular formulas would actually match this hemoresidue value? So that's first step. Understanding whether it's polar or apolar. Is this something that has some polarity or not? And this you can garner information from where is the retention time is, right? Where does it position in that chromatogram? What are the molecules that are close to that area of the chromatogram? And then MS2 fragmentation interpretation. So on one end, molecular composition. On the other end, it informs on functional groups and structure. And then from this point on, there is manual interpretation of those fragments. Literature search of MS2 never give for granted that all the MS2 are in the databases, because many of those are just published in publications. So that's literature is still very useful when it comes to understanding fragments and unknowns. And fragment similarity search. Fragment similarity search is, we saw it before with the fragment search, right? For fragments that are known for the spermidine example. Fragment similarity search is an evolution of that concept where the search will look for fragments that associate by similarity to the ones that are known. And even though it doesn't tell you this fragment is corresponds to this structure, it helps you to form hypotheses on that. And the similarity search, you can perform it in Metlin as a database that I showed before, but also you can look into fragmentation trees based on similarity search in GNPS. Then all of this will lead you to a structural hypothesis, right? Like not always happens, but it can happen. And then from there, you do either isolating and collecting enough fractions or standard synthesis, and then validating through running that standard through LCMS, spiking your sample with your standard to see whether that exact peak is the one that increases in abundance, right? Or whether so what is the behavior in the in the same LCMS system that you had when you use the standard as opposed to your analyte, NMR analysis, and the spike sample. So this workflow has bottlenecks, and that's the arrows here is just for you to have an idea where the bottlenecks are. And the reason is because I think in terms of experimental in the lab, prior that you decide to embark in something, you want to have an understanding of what type of resources and how confident you should be in that signal in order to actually pursue that. Right? Like, so green part, all the MZ database match, knowing that is an unknown, and like for metabolites that are known, like confirming them with commercial standards, that's all great. But from that moment on, really like going from an NMS2 to a structural hypothesis and a standard synthesis, this is a tough, that is a tough endeavor. So it's time consuming, and you may want to have many people with different expertise that look at your data and help you understanding whether that is a real metabolite, whether it's even possible to, the information that you have on the MS2, is it enough to actually identify that structure? Like, or at least making an hypothesis, because sometimes you have too many degrees of freedoms for those fragments, and you have not enough information, and sometimes you have to change direction. It's a matter of resource allocation. Okay, so this brings us to our training. So I will stop the recording for now.